Thanks, Elise. So Hugo told us about his team's work to understand previously unknown genes, to understand disease mechanism, and next Tim is going to tell you about the state of the art in non-coding variation, but I have the easy job, which is to talk to you about genes for which we know almost exactly what the disease mechanism is and how they function. But as Emma Farley said yesterday, we're much better at reading the genome than understanding it. So I made this plot of the ACMG 59, and on the x-axis I use registered genetic tests as a proxy for how much testing for each gene is going on, and on the y-axis you have total missense variants that are in ClinVar. And the ACMG 59 represents our best chance, these are actionable genes, so they represent our best chance at practicing genome-guided medicine right now, because if a pathogenic variant is found in one of these genes, a clinician can take action to reduce its harmful effects. I animated this plot to show you as we sequence more, we find more. This is the mo money, mo problems plot of genomics. And you'll notice that the bubbles keep climbing up, and they keep getting bigger, which means we can find more variants of uncertain significance with the more we sequence. And you'll notice they keep going up, and the bubbles never get smaller. So if I stop it at 2019 and put labels on some of these genes, you can see that they're pretty famous genes: BRCA1 and 2, the mismatch repair genes that underlie Lynch syndrome, fibrillin. They're extremely well-studied genes, and yet there are thousands of missense variants that may affect splicing or the biochemical activity of the protein, but we don't know. So they become variants of uncertain significance. And these represent a missed opportunity to either treat patients that might need help or to reassure them that they're actually not at risk for disease. There are multiple strategies to determine the disease risk of the variants. On the left there, you can see classical genetics, right? So co-segregation of the variant with the disease and families represents the gold standard. And this has been enabled more recently by direct-to-consumer genetics and by social media, but it still doesn't scale to the problem. Data sharing represents another strategy. We live in an amazing era where there are hundreds of thousands of genomes that have been sequenced and aggregated. And we can now find the allele frequency for many variants, and we can understand whether or not they must be benign because they occur at too high of a frequency to be causative, but it still doesn't help with rare variants. The good people at ClinGen and ClinVar have been using carrots to get testing companies to share their data, but most entries still don't contain enough information in order to move variants from that uncertain category to one of the more definitive categories. Functional assays represent a way to interrogate the effect of missense variation on splicing or protein function, but these assays have typically been pretty low throughput, making each variant and subjecting it to a functional assay or making an animal model. They have the power to be able to provide the information needed for variant interpretation, but they must scale to the problem. So even though this plot only represents 59 genes, there are 23,000 variants of uncertain significance in them. And they also encode proteins of diverse function, so tumor suppressors, cellular matrix proteins, ion channels, components of the sarcomere. For functional assays to represent a way forward, we have to be able to access multitude of phenotypes and functions, but at scale. So a way we could do this at scale is to use multiplex functional assays, which can measure the effects of thousands of variants across different functional elements simultaneously. These experiments are different across different functional elements, so different for promoters or enhancers or proteins, but they all consist of a few key steps. So first, we make all of the possible variants in the protein or the sequence, and we do this in a pool, and they're never separated. Then we have to get these variants into cells, and we can do this by either editing them directly into the genome or putting them in on transgenes. And next, these cells are or they don't even really have to even be cells, are subjected to a functional assay. And I left this as a black box because it's dependent on either the protein's function or your imagination. But the key is that the genotype is linked to the phenotype so that we can use next-generation sequencing as a readout to both identify and count the variants throughout a functional assay. Oops. No. We track these variants throughout a selection assay using the sequencing, and we calculate scores for each variant. So for example, we might use the ratio of its abundance after a selection assay to before to understand whether or not a variant has affected the protein's function. We then try to look at all this data together in a sequence function map. And I'm going to show you two kinds of them today. This one is when we look at protein amino acid swaps. So in this case, along the x-axis are all the positions in a protein, and along the y-axis is every possible amino acid change, and the colors represent the functional effects of each mutation. This is the second kind we're going to show you today, and this is when we look at single nucleotide variants. So each column is either A, C, T, or G, and each row represents a position, in this case, an exon. Okay, so this field really started about 10 years ago when the first paper showed that we could actually accurately measure the effects of all possible amino acids on a 30 amino acid WW domain. We could tell which mutations were tolerated and which ones weren't. And this was pretty exciting because we figured if we could scale this up and access more interesting phenotypes, we might be able to apply this to human genetics to look at genes like the ACMG59, pharmacogenes, where you might want to understand whether or not a missense variant was going to make a patient more susceptible to an overdose, or cancer drug targets, where you might want to understand whether a patient was going to have primary resistance to a targeted therapy. If we could scale all these up, we could make an atlas of variant effect, where we would be able to actually look up the effects of these missense mutations before they were found in a patient. In the past few years, according to Fritz Roth at the University of Toronto, the field has queried about 150,000 variants, which is actually on the order of the number of variants that are in ClinVar right now. Okay, so I'm going to tell you a short story about some work that we recently published, and to show you that I think these assays are kind of ready for prime time and ready to help interpret variants. So as I mentioned before, there are a lot of variants of uncertain significance in BRCA1, and this is not for lack of effort in the field. You can see in gray here that they outnumber all of the actual classified variants. As it turns out, BRCA1 is essential in haploid cells. BRCA1 is required for double-strand break repair, and one might imagine why a haploid cell line might be sensitive to loss of a protein that's important for genome integrity. So when you transfect a haploid cell line with Cas9 and a guide RNA against an exon in BRCA1, you can see on the right-hand side there that most of the cells just die. But on the left side, when you target a control gene that's non-essential, the cells are just fine. So we hypothesized that we could use a survival assay in this cell line for BRCA1 function. And so what we did was we used saturation genome editing to install all of the variants into the exons of BRCA1 using CRISPR-Cas9. And then we could just wait to see which variants were damaging and which ones could cause cell death. And we could track this by DNA sequencing. We targeted about 4,000 variants across 1,300 nucleotides of BRCA1. And we were actually able to calculate high-quality functional scores for about 3,983 of them. For the most part, the variants behaved as we would have expected, so it was nice to see that the stop codons were uniformly bad for function, that they're there on the left-hand side in red. In light blue are the synonymous variants, which were mostly functional, unless they affected splicing. And because we had edited these right into the genome, we could assess the effect on splicing. So we could see that this canonical splice variants on, mutations that hit the canonical splice bases were almost uniformly bad for function, whereas mutations farther into the introns had no effect. The missense variants, they're in green, were bimodal, as would be expected. We were able to classify these as either functional or non-functional by whether or not they behaved like synonymous variants or they behaved like the nonsense variants. Detailed functional information can be described in the sequence function map that looks like this. This is exon 2, and this blown up piece is the splice donor of exon 2. And every nucleotide position is a column. Every row is either the wild type base or the change, the single nucleotide variant. The filled in squares are the non-functional variants, and the colors of the boxes represent which variant class. So either the greens are missense, and those yellow ones are the canonical splice bases. If there's a red slash in there, it means that the RNA was depleted from day 5, so that you can, this map gives you not only the functional base, the functional effects, but also the mechanism by which the function was lost. And this is the same detailed functional map for the other 12 exons that we targeted. Importantly, using these functional cutoffs modeled on the performance of stop codons and synonymous variants, we were able to accurately discriminate between known pathogenic, which are in red, and benign variants, which were in blue, for very high specificity and sensitivity. Functional scores from this assay were able to separate 90% of the variants of uncertain significance and the variants with conflicting reports of pathogenicity into functional categories, and we're hopeful that these scores are useful for reinterpreting these variants. As I said, the goal is a sequence function atlas for all disease-related genes. In the end, this book would be 4,000 pages long, but we're starting to finally fill it in, and I'll give you a brief overview of what's going on in the field. So I just told you about our survival assay for BRCA1. Groups at the Broad and the Weizmann Institute recently published sequence function maps for P53 using survival assays in the presence and absence of drugs. Yeast can be a really handy tool. Fritz Roth's group in Toronto have taken advantage of human genes that complement their yeast ortholog to perform survival assays at huge scales for genes that cause Mendelian disease. Another group has used a humanized yeast assay to assess PTEN activity. Reporter assays in combinations with fluorescent activated cell sorting have been exploited to great effect as well. Here, a group at the Broad used a transcriptional reporter plus FACS to assess the effect of mutations in PPAR gamma, a gene important for lipodystrophy. We attempted to make a general reporter of protein stability by hooking proteins to GFP and then sorting them for green fluorescence by FACS. We created stability maps of PTEN and TPMT, which is a pharmacogene, and we were able to correctly identify up to 80% of pathogenic variants just by looking at this one protein stability phenotype. Okay. So I showed you where we are, and I just want to mention some benefits of multiplex functional assays. So one of them is, it might be obvious, but I think it can't be overstated, and that's that all of the data is actually in one place, right? There's no hunting through multiple papers from multiple labs to find variant effects of different variants. And this also is important because it means that there's no lab-specific batch effects. There's comprehensive coverage. So as I said, we aim to make every possible amino acid change or every possible nucleotide change so that the variant scores might be available before they're actually found in the clinic. These assays are also internally controlled, right? So there's every possible variant. So there's stop codons, there's synonymous variants. These should behave as expected, and if they don't, you can kind of do a little quality control of your own on there. And they're continuous and quantitative scores. So eventually, when we move away from binary pathogenic and benign classifications, these scores, or this aspect of these scores, might become more useful. And if the gold standard measurements are, or if the gold standard variants that are known to be pathogenic and benign exist, you can calculate sensitivity and specificity in order to help use these in moving variants of uncertain significance to more definitive categories. Okay, but there are still multiple problems. So we've got a problem, we still have a problem of scale and phenotypic complexity, right? So some existing technologies are low scale and high complexity, right? So making a mouse model is probably way more accurate to understand what's going on with human genetic variation, whereas others are very high scale and low complexity. So the deep mutational scans and massively parallel reporter assays assess very simple phenotypes, right? So transcription on, transcription off, dead or alive of a cell. But they're amenable to high throughput. So what we would really like to do is move these dots over to the right. We think we did that a little bit with saturation genome editing because we're able to assess not only protein function but splicing. But what will be the multiplex assays of the future where we can capture more complexity at the scale we need? And how will they be incorporated in clinical variant interpretation in a more structured manner? Because right now I just get random phone calls from lab medicine, which isn't exactly useful. So I'm really excited to see what the future holds for these.
Thank you. Now we're going to hear from Tim Reddy.